Hi everyone, welcome to another class on animal tissues. Till now we have discussed epithelial and connective tissue. Now today we are going to take up two more of the animal tissues, and that would be the muscular tissue, which gives us the ability to move, and also there are a number of our organs which are internally placed and they are not under the control of our will, but they also are having some kind of movement within them, and that is all provided by the muscular tissue. And last but not the least, the most important tissue which gives us the ability to perceive our surroundings, have certain feelings, and that is the nervous tissue. So let's begin our class with these two tissues. Now, the muscular tissue arises from the embryonic mesoderm. So in this way we can understand that once the animals became triploblastic, then only the muscular tissue originated. In case of diploblastic organism, as we had studied in the case of cnidarians, the epithelial cells themselves had some contractile fibers within them by which they were able to carry out contraction. Now, if we have to define a muscle, what is a muscle? So it is a machine for converting chemical energy, that is ATP. All organisms use energy in the form of ATP, so that ATP is converted into mechanical work. Now, if we list out the properties of the muscles, so muscles have excitability. Excitability means that they can receive a stimulus, which is any external change, and they can respond to it. That's what is excitability, or it is also called irritability. And once they receive a stimulus, they can either contract, that means their length will shorten, or they can extend, that means they will elongate. And other than that, they have elasticity, that means once the stimulus passes away, then they can return to their original form. That's what is elasticity. And conductivity is that the impulse which is generated can pass from one muscle to the other. That is of course not true for all types of muscle cells, but we will find that in some of the smooth muscles and cardiac muscles this property is very much present. Now, total muscles, if we see in the body, so they are contributing 40 to 50% of the body weight. So we can very well understand that the total body weight, a huge amount of it is contributed by muscle in a young healthy adult. But however, as we age, the number of muscle cells, they keep on declining, and that total mass is reduced to nearly 25% or so. And all that muscle is then replaced by inextensible fibers. Now, what does this muscular tissue consist of? So it consists of number of elongated cells which are called the muscle cells or the muscle fibers or the myocytes. Now, since the muscle cell are highly elongated cells, so that is why they are also called fibers. So when we say muscle fiber or muscle cell, that is one and the same. And the different types of muscular tissue that we have, there are three types: the skeletal, smooth, and the cardiac. So first of all, starting with the skeletal muscular tissue. Now, as the name itself denotes skeletal, it is so named because it is attached to the endoskeleton. Then it is also called striped muscles because they have number of striations. There are alternating light and dark bands, and these bands are visible when we see the muscle cell under the microscope. So that is why it is also called striated. Then these muscles are also called voluntary muscles because they can be made to contract or relax by conscious control. That means they are under the control of your will. So that is why they are also called voluntary. So that means we can combine three terms: skeletal, voluntary, and striated. So these three are all used for the same. However, when we say that they are under the control of our conscious mind, but it is with exceptions, like there are certain activities which are unconscious also, like blinking of the eye. There too we have these skeletal muscles, but they are blinking unconsciously without our effort. Same way, shivering in the cold, when we are shivering, it is the contraction of these skeletal muscles only, and they are contracting so that they are producing heat. So from this we can also come across another function of the muscle, that it helps to generate heat in the body. Other than, of course, all types of locomotion and movement are contributed by it. Now these skeletal muscles, they are attached to the bone by the help of tendons. So we can just see all these structure here. So here this is the bone. Now to this bone the muscle is attached by the help of tendons, and tendons, as we know, they are dense regular connective tissue. Now this entire structure here is the muscle, and this muscle is contributed by number of structures here, and this one structure is a fascicle. The plural is fasciculi. So we can see there are number of fasciculi which are bundled together to form a muscle. Now if we take one fascicle and zoom into it, so this entire structure is one fascicle. So within this fascicle, in each fascicle there are number of muscle cells. So this entire structure is muscle cell, and as we said, the muscle cell can also be called muscle fiber. And inside each muscle cell there are number of fibrils, and since they are part of muscle cell, we can put a prefix myo. The word myo will always be denoting muscle, so these are the myofibrils. And then inside each myofibril we will find there are number of myofilaments, which we will be taking up. Now in each of this part there is a connective tissue covering, and so each muscle is covered by a connective tissue sheath, each fascicle is covered by a connective tissue sheath, and each muscle cell is also covered by a connective tissue sheath. So let us see these components, the connective tissue components of the muscle. So starting from the inner, that is each muscle cell or the muscle fiber, it is covered by a sheath, and that sheath is called endomysium. Now as we said, number of muscle cells are bundled together to form the fascicle, and fascicle in turn is again ensheathed by connective tissue, and that is called perimysium. Then many fasciculi are bundled together to form the entire muscle, and it is covered by another connective tissue sheath, that is called epimysium. Then outer to epimysium there is again some dense irregular connective tissue which is in continuation with the tendons, and that is called the deep fascia. So in this way we see that these connective tissue sheaths are enclosing the muscle. Now let's move on to the detailed structure of a single muscle cell. Now taking up the skeletal muscle cells. Now here if we see one single skeletal muscle cell, now this cell, as we can see, it is cylindrical in shape and it is a syncytium. Why syncytium? Because it is a multinucleate structure. So these nuclei are located peripherally just below the sarcolemma. Sarcolemma is the plasma membrane of the skeletal muscle cell, and below the plasma membrane there are few fibers, so it is quite thick in comparison to the normal plasma membrane present in other cells. So skeletal muscle cell is cylindrical, multinucleate, syncytium. In the embryonic life, what happens that the embryonic muscle cells called myoblasts, so number of myoblasts, they fuse together to form one single cell. So the cells have fused, but their nuclei, they remain separate. So it becomes a syncytium, and once it has differentiated, it loses the ability to further divide. So muscle cells are not going to divide in our lifetime. As many muscle cells are formed in the fetal life, that would be the final. Now, within this cell, the cytoplasm which is filled in, that is called the sarcoplasm. So inside here, sarcoplasm is the cytoplasm, and in that sarcoplasm there are number of myofibrils. So by the nucleus being on the peripheral side, the entire space inside is taken up by these myofibrils. So we can imagine something like number of tiny cylinders inside this larger cylinder. Now these myofibrils are in turn made up of the myofilaments, and these myofilaments are again of two types, and those are the thick filaments and the thin filaments. The thick filaments are made up of a particular protein, that is myosin. And the thin filaments, they are made up of actin. The main protein is actin, but there are few regulatory proteins along with it, and those are troponin and tropomyosin. Now let us take the structure of a myofibril and then coming to the myofilaments. Now this entire structure here, this is a myofibril. And inside this myofibril, or within this myofibril, there is arrangement of these thick and thin filaments. And these thick and thin filaments, they are arranged in such a way that there are repeating units formed, and those repeating units are called sarcomere. The sarcomere is the functional unit of the muscle cell. Now, within this sarcomere, so if we elaborate this part, what we will find is that the sarcomere is the region between these two Z lines. These zigzag lines that we see here, these are called the Z lines, and these Z lines are also made up of a protein. That's a different protein, a structural protein. And within this, then what do we see? These are the thick filaments here, which are made up of myosin. So this is the thick one, and here there are the thin filaments. Now because of the arrangement of these myofilaments, there are certain light and dark bands can be seen, and in this we can take them one by one. So where there is a thick filament with overlapping thin filaments along with it, that is called the A band. So this A band stands for anisotropic. Anisotropic because of different physical properties. Why it is having different physical properties? Because myosin has a different density and actin has a different density. So all along the A band there are two different densities because of the two different filaments overlapping. Then there is a region in between where there is only thick filament and there is no overlapping zone. And because of that no overlapping zone, this area appears slightly lighter in comparison to the A band, and that is called the H zone. So H zone is what? H zone is the non-overlapped region within the myosin filaments or within the thick filaments. Then the region where there are thin filaments and those are not overlapped, that part is called the I band. So this I band, that stands for isotropic bands, and isotropic means having same physical properties because here there is only the thin filament present. Here these anisotropic bands or the A bands are the ones which appear dark while the isotropic bands are the ones which appear light. So that makes the alternating light and dark bands. The H zone in between is a slightly lighter than the A band but darker than the I band. Now let us see what happens during the contraction. During contraction, these thin filaments, they slide over the thick filaments, and that is why it is called the sliding filament theory. Now let us see what happens when this kind of sliding occurs. So in this case, when they slide over, what happens as they are coming towards the center? So what is happening? The overlapped area, the zone of overlap is increasing, and this H zone which is in between, that will be decreasing. And when there is the entire sliding has taken place, the H zone will disappear. These thick filaments do not move from their location, so there won't be any change in the A band. So in this case, we can see that once the sliding has occurred, what has happened? The length of the sarcomere has decreased, the H zone has decreased, the length of the I band, because I band was which region? It was the region which is not overlapped, but now it has gone in, so now it is the major area has got overlapped, so even the I bands have decreased. So decrease in the length of H zone, I bands and the sarcomere. What has remained still the same? That is the A band. A band has not undergone a change. Now let us take a question. Which of the following is not a characteristic feature of biceps muscles? Now biceps are one of the skeletal muscle that is over our arm. So when we flex our arm, that is when we bend our arm, that is because of the contraction of biceps. So we are usually able to make it contract merely by thinking about it. That means it is under conscious control. It has alternate light and dark bands. Its muscle fiber taper at both ends, and its muscle fibers are bundled together in a parallel fashion. So we have to consider which is the odd one here. Now since it is a skeletal muscle, it is voluntary. So the statement 1 is correct. Then skeletal muscles are striated, having alternate light and dark bands, so even that is correct. And its muscle fiber taper at both ends. Now the muscle fiber, skeletal muscle fiber, they are cylindrical. So that means this statement is incorrect, and they are of course bundled together to form the fascicle. So here the answer would be option number 3. Now let's move on to smooth muscle tissue. Now the smooth muscle tissue. Now in this case, the smooth muscle cells, they are fusiform. Fusiform means spindle shaped. They are thicker in the middle and they are tapering on both the ends, and these are uninucleate, where the nucleus is centrally placed. So we can just mention that they are having this single central nucleus. And these are activated involuntarily. That means they are not under the control of our will. And these also do not have striations. So these muscles, they can also be called unstriated. So on the basis of structure and function, we can compare them with the skeletal muscles, that these are unstriated and these are uninucleate. Even the shape of the cell is different. It is not cylindrical, rather it is fusiform. Now these smooth muscle tissue, they are categorized into two types, that is the single unit and the multi-unit smooth muscle tissue. So let us take them one by one. So first of all, the single unit. Now in this case, there are number of smooth muscle cells or the fibers which are connected to each other by gap junctions. So if we see this arrangement, this is how it is present, and here this is the smooth muscle cell. So we can see number of cells are arranged along with each other, and between the cells there are number of gap junctions. And if you remember what is a gap junction, these are the junctions which connect the plasma membranes of two neighboring cells so that there is communication taking place between the two cells. And here this is the motor nerve, the nerve which innervates the muscles, that is a motor nerve. So when this motor nerve, it releases the neurotransmitter, so with whichever cell it is having a junction, that cell will be stimulated and that cell will conduct the impulse, but that will quickly transmit to all the other cells because all of them are connected by gap junctions. And these single unit smooth muscle cells are also called the visceral smooth muscles because they are found in the visceral organs, in the walls of hollow organs such as stomach, intestines, uterus, urinary bladder, so on. And some of them are autorhythmic also. Autorhythmic means that they can undergo contraction without the stimulus from the motor neuron. So without that they can have their own rhythm, so that's why we have the term for it, autorhythmic. Now coming on to the multi-unit smooth muscles. Now here we see the arrangement. Here this is the smooth muscle cell. And again here it is the motor neuron. And in this case the cells are apart, they are not having many gap junctions, few cells may be connected but more or less they are scattered. As a result, what happens that these cells can contract individually, they are not connected. So that means number of small units are formed. Now these are located in the walls of the large arteries, in the airways of the lungs. Erector pili muscle. Erector pili muscle are one of the smooth muscle which is connected to the hair follicle, and the word pili comes for hair anyways, and when this muscle contracts there is raising of the hair as it happens when we are frightened or there is cold air outside, then in that case this erector pili muscle will contract. Iris, which is a muscle controlling the diameter of our pupil through which the light enters. Ciliary body, that is again in the eye, which is helping in the accommodation of the eye lens. So here the muscle type is the multi-unit smooth muscle. Now moving on to the cardiac muscle tissue. Now cardiac muscle tissue has the property of both skeletal muscle tissue as well as smooth muscle tissue. Now like skeletal muscles they are striated, and like smooth muscles they are involuntary. Their cell type, if we see, it is cylindrical but it is branched and it is having a single nucleus. It is uninucleate most of the times. Sometimes it can be binucleate also, that means having two nuclei, but usually it is having a single nucleus. And here the cells are attached end to end, and where two cells attach the plasma membranes are quite thick, and those thickenings are called intercalated disc. Now this structure, intercalated disc, is unique only to the cardiac muscle cells. It is not found in the rest of the other two muscle tissue. And at these intercalated discs there are present two types of cell junctions. One is the desmosomes. Now desmosomes are the adhering junction, that means which attach the cells together, because as the name suggests the cardiac muscle tissue is found in the heart, and within the heart the blood is exerting pressure on the heart wall. So because of that pressure the muscle cells should not break away, and these desmosomes are holding the cells together. And then here there are gap junctions as well, because when there is a stimulus, then that stimulus will quickly spread from one cell to the other, so that the entire chamber of the heart can contract as one unit. So that is why a lot of gap junctions are also there. So at these intercalated discs, these two important junctions we have, desmosomes and gap junctions. Now coming to the location, so as we know from the name itself that it is present in the wall of the heart, and that layer of in the wall of the heart where cardiac muscles are present, it is called myocardium. And then again these cardiac muscles, they are in our case, rather in all vertebrates, they are autorhythmic. That means they are not dependent on the motor neuron to charge them to contract, rather they can generate their own impulse. So they are autorhythmic. Although nerves certainly reach the heart, which can increase the heartbeat rate or decrease it, but the initiation is done by the muscle themselves. So these muscles are also autorhythmic. Now after taking these three types of muscle tissue, let us compare them. Now here characteristics, we are considering some of them and comparing the three of them. Coming on to the appearance, as we have seen, skeletal muscle cell, they are cylindrical, multinucleate, that is syncytium. Smooth muscle cells, fusiform, uninucleate. Cardiac muscle cells, cylindrical, branch and uninucleate. Then their location, if we see, these are attached to the bones by the help of tendons. These are present in the visceral organs, and this is present in the heart. Then coming on to striations, so in skeletal muscles striations are present, in cardiac muscles striations are present, but these striations are absent in case of smooth muscle. Then coming on to the cell junctions, in case of a skeletal muscle there are no cell junctions. Here these are present, but only in the single unit type. And here again these are present. Then nervous control, the skeletal muscles are under the control of the central nervous system, that is our conscious, from the conscious brain these are controlled, while these are controlled, these smooth and the cardiac muscle, these are controlled by autonomic nervous system. Now how does a nerve control any muscle? So it will be releasing a chemical substance that is called a neurotransmitter. So the neurotransmitter which is released here, this one in case of a skeletal muscle, it is acetylcholine. In short, we can write it as ACH. Now this acetylcholine, when it is released by the nerve cell, the skeletal muscle is stimulated to contract. Now in case of the smooth and the cardiac muscle, it is acetylcholine and there is another one, that is norepinephrine. These two have regulatory activities because the autonomic nervous system is maintaining homeostasis. So it can increase the activity of the muscle or it can decrease the activity. So accordingly, there are two different neurotransmitters used which are antagonistic to each other. One can increase the activity, the other can decrease. And in different places, they have different roles which we will be discussing in detail with the nervous system. Same way on the cardiac muscle, acetylcholine and norepinephrine. Both of them have their activity. Like norepinephrine will be increasing the heartbeat rate, acetylcholine is going to decrease it. Now let us use this information and solve a question. Now which of the following is the correct pairing of a body part and the kind of muscle tissue that moves it. Now here, iris, involuntary smooth muscle. Now just now we have mentioned that in case of iris we have multi-unit smooth muscle. So this is correct, but let us see what is wrong in the other three. Heart wall, involuntary, cardiac muscle, so involuntary, that's right, but it is striated, while here the option says unstriated. Biceps of the upper arm, this is very much skeletal muscle, not smooth. Abdominal wall, try to imagine, even if you don't know it, if you try to pull in your abdomen by your will, you can do it. So that means it is also skeletal muscle, not smooth. So that means here our answer is option number 1. Then let's take one more question. Smooth muscles are, now this is for their shape, involuntary, fusiform and non-striated. So the first one itself is the correct answer. So this much completes about the muscular tissue. Now let's move on to the nervous tissue. The nervous tissue, the tissue which gives us the ability to feel and to respond to various stimuli. Now, if we see nervous tissue under the light microscope, this is how it will appear, in which these cells, from which number of cytoplasmic processes will be arising, and these cells are the structural and functional unit of the nervous tissue, that is the neuron. And another cell which are more abundant than the neurons, and those cells are called the neuroglial cells, or simply neuroglia. These are also called glial cells. So these are the two cells which constitute the nervous tissue. Now as far as the nervous tissue is concerned with the origin, it is ectodermal in origin. Now the neurons, these are the longest cells of the body. So that's why these are also called the nerve fibers. So neurons, nerve cells, nerve fibers, they mean one and the same. And they are specialized non-dividing cells, because they are so specialized that they have lost the power of division. So they are not dividing at all, and they have the special powers of irritability. That means they can sense and respond to the stimulus, and not only that, all along the length of the neuron the impulse will be conducted and then it will be passed on to other nerve cells. Now this ability of generating and conducting electrochemical impulses is the characteristic of the neurons. So that is why it is termed excitable cell. Now if we see one single nerve cell, so this nerve cell comprises three structures: the cell body, the dendrites and the axon. Now let us see this in a particular nerve cell. Now here this is one single nerve cell, and it is much longer than it is actually represented over here. So this structure here that we see, this is the cell body or the cyton. and in this cyton there is quite large nucleus, and there are certain granules which are seen, which is again a characteristic of the nerve cell, and those are called Nissel's granules. Now these Nissel's granules are actually the rough endoplasmic reticulum. So rough endoplasmic reticulum is the reticulum over which there are ribosomes present. So it has RER and ribosomes. Then from this cell body there are number of processes arising. These tiny branch-like processes which are arising, these are called the dendrites. These are the dendrites, and one single long process that is called the axon. The region from where the axon arises, that is called axon hillock, and it is the integrating center because the various stimuli are received by the dendrites. Now these impulses can be excitatory or inhibitory, but at the hillock these will be integrated, that is excitatory and inhibitory. There will be summation so that whether an excitatory impulse has to pass through the axon or inhibitory, that will be determined at this hillock. So it is the most sensitive part of a neuron. Now from this axon, if we see over this, there are layers and layers of some material over it, and that is actually the myelin sheath. Now this myelin sheath is made up of myelin, which is a kind of lipid. It's a lipid, and the purpose of myelin is that it is insulating the axon. So wherever myelin is present, the ions cannot move across the axon membrane. The axon membrane is called exolemma. So through that the movement of the ions will not occur if myelin sheath is present. But we will find there are regions where myelin is absent, and those regions are called the nodes of Ranvier. So the node of Ranvier is the region from where myelin sheath is absent. Now this myelin has been formed by the Schwann cell. The Schwann cell has wrapped around the axon and invested the axon with myelin, so that the cytoplasm and nucleus of the Schwann cell itself has become peripheral. And this layer of cytoplasm of the Schwann cell, that is forming the neurolemma. So if we go from inside to outside, we will see that axon is filled with the cytoplasm, which is called exoplasm, and the exoplasm is different from the cytoplasm present in the cyton, in the sense that here the Nissel's granules are absent. Now this exoplasm is surrounded by exolemma, then outer to exolemma is myelin sheath, and outermost is the neurolemma. And neurolemma, as we said, is the layer of peripheral cytoplasm of the Schwann cell. So here we see the nucleus of the Schwann cell, so we can mark it as well. Then at the node of Ranvier we can see another fiber is arising. So these are the branches coming out from the axon, and such fibers which are arising at right angle from the axon, from the nodal area, these are called collateral fibers. So from one axon there can be many collateral fibers. Now where the axon ends, it again forms number of branches. Now these branches, they are ending in a knob-like structure which is called the synaptic knob, and this branch structure here is called axon terminal or telodendria. Now this type of neuron is usually represented in the diagrams, and this neuron which we are seeing here, it is having many dendrites, so that it means that it can receive stimuli from various directions, but of course axon will always be one. Now such type of neuron is called multipolar neuron. So this neuron is multipolar neuron. So accordingly we can categorize the various types of neurons, and if you can recall that the nerve cell for the first time appeared in the phylum Cnidaria, and there that particular nerve cell is the most primitive type, that is called apolar or non-polar. After taking up the structure of the neuron, let's take up a few terms which we will be using time and again. Now first of all, in the nervous system we know that there will be two major system divisions that we will be studying: the central nervous system or the CNS, in which we take the brain and the spinal cord, and the peripheral nervous system, in which we will take all those nerves which are connected to the brain and the spinal cord. So in these two systems we will be using different terms. One is the ganglia, which is a group of cell bodies. Cell bodies, that is the cytons, a cluster of cytons together, that is called ganglion, but that term will be reserved for the peripheral nervous system. And for the same structure in the central nervous system we will use the term nuclei. Then similarly, when there are number of axons together, then that is called nerve, but that term again is reserved for peripheral nervous system. The same structure in the CNS will be called tract. Now just like in case of muscle tissue, we had seen that number of muscle cells are bundled together to form fascicle, and many fasciculi are grouped together to form a muscle. Same goes in the case of nervous tissue as well. So each neuron is surrounded by a connective tissue sheath that is called endoneurium, and then many axons, they are bundled together to form the fascicle, which is covered by perineurium, and then many fasciculi together are grouped to form the nerve or the tract, which is covered by connective tissue sheath called epineurium. So this is the connective tissue sheaths which are covering the entire structure. Now where one nerve cell ends, it transmits the information to the next nerve cell. So the site of contact between two neurons is called synapse, and at the synapse the neurons are not in touch with each other, rather there is a narrow gap in between, which is called the synaptic cleft. Now these synapses can be of various types, and their name itself shows what different structures are making the contact. So the most common ones are exodendritic. That means the axon of one neuron is making the contact with the dendrite of another neuron. Similarly, exosomatic, that is axon of one with the soma, that is the cyton, cell body of the other neuron. And exoaxonic, these type of synapses are found in the brain, that is axon of one neuron with the axon of the other. So these are the various types of synapses. Now let us take a question. Now 4 healthy people in their 20s got involved in injuries resulting in damage and death of few cells of the following. Which of the cells are least likely to be replaced by new cells? That means it is, the question asks whether the cell will be able to divide or not, because only cell division can replace the lost cells. So osteocytes, these are the bone cells, and as we know, in the bone there are the osteoblasts which are continuously dividing and forming osteocytes, Malpighian layer of the skin, that is the stratum germinativum, that is a mitotically active layer. Liver cells, they are also mitotically active. Neurons. Neurons are certainly not going to divide, so this is the right answer. Then another one, the Nissel's granules of the nerve cells are made up of. So in this case, Nissel's granules, as we said, is rough endoplasmic reticulum, and on this rough endoplasmic reticulum there are ribosomes present. So here, out of the 4 choices, this would be the best answer. So option 3 would be the right answer. Now let's move on to the different types of nerve cells. Now there are various types of neurons. The neurons for the first time appeared in the animal kingdom in the phylum Cnidaria. And if you recall, the type of neurons in case of cnidarians were apolar or nonpolar. So first of all, we are starting with this apolar or nonpolar. And in this case, we can see there is a cell body, and from this cell body there are number of cytoplasmic projections arising, but they are not distinct dendrites or axons. All processes are similar, so we can say they are undifferentiated. And as a result, they can receive stimulus from any end and they can pass it on to any direction. And that's what makes their nervous system as a diffuse type. So this type of neurons we will find in case of cnidarians. Then next one, unipolar. In case of unipolar, here this is the cell body, and from the cell body there is a single process arising, and this process is the axon, and there are no dendrites at all. So that is unipolar. Now this type of neuron is found in embryo. And also among non-chordates, but among vertebrates if we talk about, then it is only at the embryonic stage. Then another one, bipolar. In this bipolar neuron, this is the cell body, and from this there are 2 processes arising. One is the dendrite, the other one is the axon. So there are 2 poles, and that's why it is bipolar. And this type of neuron we will find in our retina, olfactory epithelium, then the most commonly studied neuron, that is multipolar, which just now we had mentioned. So here there are number of dendrites and 1 axon, and they are abundantly present in our central nervous system, that is the brain and the spinal cord. And last but not the least, the pseudo unipolar. Now the word pseudo means false or fake. So it is appearing to be unipolar because from the cell body a single process is arising, but it is actually bipolar because this process is bifurcating into 2. One of it will be the dendrite to receive the stimulus, and the other one would be the axon. Now this type of neuron we find in case of dorsal root ganglia, of spinal cord. Now moving on to the neuroglial cells, that is the cells which are more in number in the nervous tissue, but they do not conduct impulses, and these neuroglial cells are dividing cells. So when we study nervous system, all the limelight is focused on the nerve cells, and the glial cells are supposedly the silent partners, but they are the ones which are providing nourishment to the nerve cells, removing the waste, and at times helping in repair. In the fetal life, they even secrete the nerve growth factors by which the nerve cells divide at that time, and they also form the myelin sheath. So there are a number of housekeeping functions that these neuroglial cells are carrying out. Now these neuroglial cells are present in the CNS as well as in the PNS. In the central nervous system, 3 types of neuroglial cells are present, and those are oligodendrocytes, astrocytes, and microglia. Now these oligodendrocytes, so here this is the cell, and from this cell there are number of cytoplasmic processes arising, and these processes are wrapping over the axon, and that is why insulating with myelin. So in case of CNS, when myelin sheath is present, that myelin sheath will be formed by oligodendrocytes, not the Schwann cell. So this is in the CNS. So all myelinated axons in the CNS are covered by the oligodendrocytes, and one oligodendrocyte can ensheath number of different axons. Then the other one, that is the astrocytes. Now astrocytes, this is cell here, this is the astrocyte, and here this is the nerve cell. So here it is the neuron. This one is the astrocyte, and here this is the blood capillary. So what do we see here? That from the astrocyte there are number of, again, cytoplasmic processes which are covering the capillary. So over the surface of the capillary they are forming tight junctions with the endothelium, and as we know, when there is a tight junction, then substances cannot leak out. So as a result, the permeability of the capillary is decreased, and many substances are not allowed to pass to the neurons. So this is what is called the blood-brain barrier, and this is important because nerve cells are very, very delicate cells, so there shouldn't be sudden fluctuation of any substance. A certain barrier is required, and that is why any kind of medicine which is given specifically for the nerve tissue, it is made in such a way that it should be able to cross the blood-brain barrier. Then moving on to the third type, that is microglia. Microglia are phagocytes. They are phagocytic cells, and these are the only cells in the nervous tissue which are mesodermal in origin, because they are the resident macrophages, and these are mesodermal in origin. And last but not the least, the Schwann cells. Schwann cells are found in the PNS. So these are the cells which form the myelin sheath in the peripheral nervous system. So in case of CNS, oligodendrocytes, astrocytes, and microglia are present, and in the PNS only one type of glial cell is present, and that is the Schwann cell. And these Schwann cells, as we said, that their cytoplasm on the peripheral side, that forms the neurilemma. So that is why these cells can also be called neurilemmocytes. And they are different from oligodendrocyte, that is one Schwann cell covers only one axon, while one oligodendrocyte covers many axons. And in case of oligodendrocyte, there will be myelin sheath formed, but there won't be any neurilemma. Now let us take a question here. Unipolar neurons with an axon and no dendrite are present in. So unipolar neurons, if we see these choices, in the retina we have bipolar. In the brain, maximally multipolar. Dorsal root ganglia, it is pseudo unipolar, and of course it is the embryo where unipolar neurons are present. So here answer 1 is the correct answer. So this brings us to the end of today's class, and with this we have completed the topic of animal tissues. In the next class we will take up the morphology of animals. That's all for now. Thank you, students.